Next we have the cytoskeleton. Now we are talking about the cell and we have to talk about the skeleton of cell. Now you know what is the function of skeleton? Skeleton provides the shape, basic shape to the body. We know that. We study it in the skeletal systems, and over there we say that one of the important functions of the skeleton is providing the shape. Otherwise the body, considering our body, if we say, it would be a lump of meat basically. In the case of cell, we have cytoskeleton. As you can see over here, we are talking about cytoskeleton. We are going to break the word, and we have cyto, which refers to cell, and we have skeleton, which refers to the skeletal components of the cell. Now there won't be bones, but there would be something which would be providing the shape to the cell. And when we talk about the cytoskeleton, we deal with different type of filaments. Now those filaments are polymeric proteins only. They are proteins which form some sort of filament or a tubule which would provide the shape to the cell. You know, cell is a three-dimensional structure and it is going to have certain shape of it, and that shape would be provided by those particular filaments or the cytoskeletal elements. Now we will not get into depth of what is cytoskeleton. We just have to understand that what is the composition of cytoskeleton. That is more important. Instead of understanding that what is cytoskeleton, how it would look like, this is one single representation where we are considering the part of cell. Cell is basically an ellipsoid body. Consider it as a sandwich. Consider it as a sandwich, and all the particular organelles are sandwiched inside the two plasma membranes. As you can see over here, there is mitochondria, there are ribosomes, rough endoplasmic reticulum. What you see over here is that there is a meshwork of filaments. The cytoskeleton is forming a meshwork so that both the plasma membranes, the gap between the plasma membranes is intact and it does not fall on the organelles. I hope you are getting my point. Like supposedly, if you have to make a tent, you would provide it a little bit of framework of some sort of hard substance so that the tent does not collapse in itself. So that framework would be providing the skeleton to the tent. In the same way, these particular filaments which are formed as a result of polymerization, oftenly they are homopolymers or heteropolymers of a specific protein that we study. These particular filaments, they are providing that framework to the cell, and hence they are known as cytoskeleton. Now enough said about it. Nothing more is to be understood because this is the simple definition which I have extended so long. What you have to understand is that there are three types of cytoskeletal elements. First one is microfilament, and as I am telling you one by one, please pay attention. It is according to, in their increasing order, according to their size. As you see over here, the microfilaments, as the name suggests, micro, that means smallest. Those filaments which are smallest in size are microfilaments, and they are formed by polymerization of actin monomers. There are two different chains which are formed and then they get coiled. Supposedly you are having two beaded strings and then you coil them, and those beads are all of similar color. So you have actin monomers. This is a homopolymer of actin which is present in the form of microfilaments inside the cell. Then we have, next is the intermediate filaments. That means this one was the smallest. The one which lies between the largest in size and the smallest in size would be intermediate filament. And intermediate filament is just like, supposedly you have a pen stand and you fill it completely with all the pens. You can picturize that thing. Supposedly you are holding all the pens in your hand like this, and then they are, consider I am having 10 markers like this, and all the 10 markers are completely in a column and they are clustered together. Then it would form the intermediate filament. Over here you see the diameter basically is 18 to 12 nanometer, which is greater than that of microfilament, which was 7 nanometer. These points are to be learned because they can be important from exam point of view, from learning point of view, they are important. There is a fibrous unit which is being formed, and the intermediate filament is the result of this polymerization. Then the last one is the microtubule, and as you can see over here, these two are heteropolymers. Here you see the monomers are of different colors. So we have a tubulin dimer, beta tubulin monomer and alpha tubulin monomer. So the microtubule, as you can see, it is having a hollow core. There is a hollow core. Microtubule is more like a tube structure where the tubulins are arranged on the periphery. Supposedly now you have a container or a jar, or you can say a glass, and you put all the beads around the periphery. Then what you have is internal hollow chamber, and then there is beads which are surrounding. So that is microtubule, and as you can see, there is this is almost double the size of intermediate filaments and 3 times that of the microfilaments. So these are the 3 different cytoskeletal elements, and you have to pay attention that these microtubules are very important because they form the basis of formation of flagella and cilia and centrosome. Of course, they are formed of microtubules only. Now one by one we are going to see what these cytoskeletal elements are all about and what they have in their specific features. So first of all, what are microfilaments? They are the polymers of actin. Actin is a protein. They are actin filaments. They are the thinnest class of fibers. This particular thing is repetition of what I have said earlier. They are the thinnest of fibers. They are made up of actin protein, which is a solid rod, that means coiled one. The twisted double chain of actin subunits. Yes, I told you that you are having 2 beaded strings and you have to coil them accordingly. So there is a twisted beaded chain, okay, double chain of actin subunits. So there is an actin monomer, there is a monomer, actin, then there is a long actin subunit, there is another actin subunit, and they get helically coiled and they form a particular polymer, and we have microfilament as a result. The diameter is about 7 nanometer, already discussed. What is the function? Function is that they provide that framework, 3D framework, 3-dimensional framework of the cell would be formed. In muscle cells, actin filaments interact with myosin filaments to create muscle contraction. When we study human physiology, we have actin myosin interaction which takes place, and as a result of actin myosin interaction, there is a sliding filament theory which brings about contraction. This is, if you have already seen what muscle contraction is about, you will be very easily understanding what it is about. If you don't know, you need to see that video and you will be quite clear that how actin and myosin are both interacting. So the actin filaments, that are microfilaments, they interact with those of myosin and bring about the contraction. So that is one important function that these microfilaments perform. These specifications are quite important to learn. Now the next one is the intermediate filaments. Over here, as we had actin, in the case of microfilaments, we have keratin proteins over here, okay. The keratin proteins are the same as we have in our hair, body hair. So it is built from keratin proteins. They are important for the bearing of tension, whatever tension is present inside the cell. You know, whenever there is a framework being formed, in case you are building a house, there is something in order to bear the tension, and we have different architectural specifications in our houses that we make. So why not in a cell? So that part of the cell which is specialized for bearing the tension, that is known as the intermediate filament, which has the main function of tension bearing. Apart from that, the size is 18 to 12 nanometer, already discussed. It is made up of keratin protein, and come to its structure, there are protein subunits which are coiled in all together. Together they are coiled, and there is no hollow space in between, that you have to remember. The function is they hold things in place inside the cell. Yes, they are going to do that. They are more permanent fixtures of cytoskeleton. As I told you, they are specialized for bearing tension, so they would be more permanent as compared to those of microfilament. They reinforce the cell shape and fix organelle location. So basically you have different types of mortar and pestle that we have concrete sort of thing in our constructions. This part is working as a concrete arrangement of the cell is being brought about by the intermediate filaments, and the nucleus is held in place by a network of intermediate filaments. This is an important function of intermediate filaments, that They are important for reinforcing the structure of cell. That means whatever hard part of the cell, that means whatever weight is to be borne, whatever tough framework is to be provided, that would be provided by intermediate filaments. Next we move to the microtubules. Microtubules, as you can see, it is made up of tubulin. Now tubulin is also a protein. There is a dimer, that is alpha tubulin and beta tubulin are present. Dimer is formed and then the dimer undergoes coiling, leaving a hollow space which is around 25 nanometer. It is just like a spring. You have seen a spring, or you take a thread and you coil it over, you take a beaded string, I would say. One bead would be of alpha tubulin, other would be of beta tubulin, and you coil that beaded string on a tumbler supposedly, or you coil it on this marker itself. Then you take out the marker from there, supposedly you are coiling it like this, and then you take out. There would be a central space that would be left inside, and you will have the tubulin dimers which would be surrounding it, just like a spring. How do you form a spring, or how do you form a solenoid? In the same way, a microtubule is being formed, where we have tubulin dimers, so many tubulin dimers, and they coil accordingly, and they leave a 25 nanometer gap in between them. That is what a microtubule is made up of. It is the thickest fiber. It is a hollow rod of about 25 nanometer in diameter. Yes, it is constructed by a protein tubulin, which is having two different forms. It grows or shrinks as more tubulin molecules are added or removed. As much as tubulin molecules would be added, the diameter is going to increase. No, the diameter is going to remain similar. It is going to increase in size. I am repeating it again. This statement might confuse you a bit. It grows or shrinks as more tubulin molecules are added or removed. It is not going to decrease in diameter. Please remember, the diameter would remain 25 nanometer. It is similar as you are making a solenoid. You have this much thread, or you had this much wire, so it is going to form this much solenoid. Supposedly the wire is increased into this length, so the solenoid would be larger in size. It will not change in the diameter around which ever coil you are doing it. The diameter won't change. So this is all about microtubules. Now, because we are talking about microtubules, let us see what it is formed about. A little bit better understanding for this I have used it. This is the heteropolymer. So we have alpha and beta tubulins, heterodimers. Both of them, they come close to each other and they form the protofilament. This is alpha, beta, these are tubulin. There is one negative end at which we have alpha tubulin, and there is a positive end at which we have the beta tubulin. Then what happens is, this negative end and this positive end, they coil accordingly, one by one they are going to coil, and we'll have one negative end and one positive end. At the negative end there would be tubulin alpha tubulins, and at the positive end there would be beta tubulins. So we have a protofilament which gives rise to different microtubules. The positive and negative end you have to keep in mind, and in this way we have a microtubule which is formed, and please remember the diameter remains 25 nanometer itself. So this is all about the cytoskeleton of the cell, which provides it the basic framework so that the cell does not collapse on itself. The two plasma membrane have a basic meshwork, or you can say the filamentous network, which is providing it the cytoskeletal properties. We have microfilaments, we have intermediate filaments, and we have microtubules in it.